Welcome back to the Integrated Neurobiology, Structure, Function, and Pathology course. I am incredibly excited that you are here with us today.
It's great to be back.
If you've been following along with the syllabus, you know exactly where we are. But for everyone tuning in, today we are taking a deep dive into a really pivotal session. This is Lecture 3, Neuropharmacology Principles.
And you're absolutely right to frame it as pivotal. It really is the bridge.
The bridge?
Yeah, I mean, if you recall, in Lecture 2, we spent that entire hour deep in the biophysics. We were talking Nernst equations, membrane potentials, all those calcium channels and fusion pores.
Right, the hardware, the nuts and bolts of how a signal gets from A to B.
The hardware, exactly, the wiring. Today, with Lecture 3, we're moving on to the software. We're shifting from the physics of the synapse to the chemical manipulation of those systems.
And this is the last piece of Module 1, right?
Yeah.
Cellular foundations and neurophysiology.
It is. This lecture wraps up the foundational science, which, it perfectly sets us up for the major shift that's coming next time.
Where we finally zoom out. Lecture 4 is when we start mapping the actual anatomy, the embryology.
We do.
Yeah.
But I always tell my students, and this is so crucial for the physician scientists listening, before you can appreciate the map of the city, you have to understand the traffic signals.
You need to know what red and green actually mean.
Precisely. So the mission for this deep dive for today is to really synthesize the principles of how these neurotransmitters are made, how they act on their receptors, and maybe the most important part, how those signals are terminated.
Because if you can't stop the signal, it's just noise. You can't send a new coherent message.
It's just noise, exactly. For any clinician listening, this lecture is, it's essentially the instruction manual for your toolkit.
The toolkit, I like that.
It's the absolute foundation for understanding every single drug class we will discuss later in this course.
Yeah.
I'm talking antidepressants, antipsychotics, anxiolytics, mood stabilizers, all of them.
They all work by tweaking these specific systems.
Every single one. You just cannot prescribe rationally or safely without a deep understanding of these core mechanisms.
Okay, I love that framing. So we have 15 slides to get through today, and I know we want to stick strictly to that slide order to make sure everyone can follow along for the exam and for clinical practice.
That's the plan.
So let's not waste any time. Let's jump right into Section 1, Systems Organization and General Principles. We're looking at Slide 1 now. Title and learning objectives, hierarchical versus diffuse.
Right. So slide 1 immediately sets the stage by differentiating the two main organizational structures in the central nervous system.
And this is something I think people miss. We sort of picture the brain as one big homogeneous computer.
And it's not. It operates using two very different, very distinct wiring schemes.
The slide shows this really well with the split diagram. On the left side, you've got what looks like a very precise hardwired connection, point A to point B.
Mm-hmm.
And on the right, well, it looks a lot more like a sprinkler system.
That is a perfect analogy. Really it is. The first type, the one on the left, that's the hierarchical system.
Okay.
Think of your sensory and motor pathways. They are incredibly precise, they are fast, and they are strictly point to point.
So like touching a hot stove and pulling your hand away.
That is the classic example. The information travels from a specific receptor on your skin to a specific neuron in the spinal cord, up to a specific spot in the thalamus, and then to a specific patch of cortex.
So zero ambiguity. Input A always leads to output B. It's linear.
It has to be. These systems use large myelinated fibers because speed is everything. Conduction velocity is king here. You have to react to the world in real time.
And what are the chemicals involved?
The major players in this hierarchical world are glutamate for excitation and GABA for inhibition.
The on and off switches.
That's all they are. They are transmitting specific bits of data. It's digital, on or off, yes or no.
Okay, so that's the hardwiring, the Ethernet cable of the brain.
Yeah.
What about that sprinkler system on the right side of slide 1?
So those are the diffuse systems. And their job is totally different. They're modulatory. The neurons here, they often originate in these tiny specific nuclei deep in the brainstem.
But they project everywhere.
Everywhere. The axons branch profusely. I mean, a single neuron from a place called the locus ceruleus can have an axonal network that covers huge areas of the cortex, the cerebellum, even the spinal cord.
And they don't form those neat one-to-one synapses.
Not always. As that diagram shows, they often release their neurotransmitter from multiple little swellings along the axon called varicosities.
Varicosities, you mean they just kind of leak it out?
It's an intentional leak. Think of it like beads on a string. As the action potential travels down that axon, these varicosities just sort of spray the neurotransmitter into the extracellular space around them.
There's a term for that, isn't there?
There is. It's often called volume transmission.
Volume transmission. Okay, that sounds so much messier than the point-to-point stuff. It sounds like painting with a spray can instead of a fine point pen.
It is messy, but it's incredibly efficient for a different purpose. These are the systems that use the monoamines. Dopamine, serotonin, norepinephrine.
Right.
They aren't telling your finger to move left or right. They're setting the overall tone or the gain of the brain.
So they're determining whether you're awake or asleep or anxious or depressed.
Exactly. They modulate the entire environment in which those hierarchical point-to-point systems have to operate.
So if I can try to summarize this, hierarchical is for the data, the actual content of a thought or a movement. And diffuse is for the state or the mood, how you feel about that data or the context it's in.
That is the perfect distinction. And if you look at the learning objectives on this slide, that's our roadmap. We're going to map the rate-limiting steps for making these transmitters and, crucially, compare how they get cleaned up. Termination.
Because how you clean up the mess determines how long the signal lasts.
And how strong it is. It's everything.
All right, let's move on. We'll skip slide 2, the overview, and go straight to slide 3. This gives us the visual framework for that cleanup. It's titled The Life Cycle of a Neurotransmitter. It shows a nerve terminal with 5 steps numbered 1 through 5.
This is what I call the 5 targets concept, and I cannot stress this enough for everyone listening.
Okay.
Every single pharmacologic intervention we use in the central nervous system hits one of these 5 steps. If you truly deeply understand this one slide, you understand the mechanism of action for almost every psychoactive drug out there.
So this is the Rosetta Stone for the rest of the course.
It really is. It provides the logic.
Okay, let's walk through the numbers then. Target 1 is synthesis.
Right. The neuron has to actually make the transmitter. This usually happens in the cytosol with the help of enzymes. It's a factory floor.
So if you block an enzyme,
you run out of product. It's a supply chain issue. No raw materials or no workers. The enzymes means you have nothing to ship out.
Simple enough. Target 2 is storage. Now, why is this a separate step? Why not just make it and release it?
This is such a critical and often overlooked nuance of neurobiology. You can't just have these neurotransmitters, especially the monoamines like dopamine or serotonin, just floating around freely in the cytoplasm.
Why not?
Well, for one, they're chemically unstable. But more importantly, the cell actually has enzymes like monoamine oxidase, just sitting there on the mitochondria waiting to chew them up.
As a protective measure.
Exactly, to protect the cell from oxidative stress.
Yeah.
So you have to package them, you put them in these little lipid bubbles called vesicles, both for protection and for regulated release.
And the slide labels the transporter that does this as VMAT, the vesicular monoamine transporter. And I'm seeing something about a proton gradient here.
Yes, VMAT is a marvel of biological engineering. It's an active transporter. The cell first uses an ATPase to pump protons into the vesicle. Then VMAT uses that gradient. It lets one proton out in order to pump one molecule of dopamine in. It's pushing against a massive concentration gradient.
So it's really cramming them in there.
It's like stuffing a jack-in-the-box. You're packing thousands and thousands of molecules into this tiny sphere, coiling the spring, just waiting for the trigger to pop it open.
Which brings us to a specific drug mentioned on the slide, Reserpine. Now, I've heard of this, but it's pretty old school, right? We don't really use it anymore.
Rarely, if ever, in modern practice. The side effects are just too severe. But as a teaching tool, its mechanism is the perfect prototype of a storage blocker.
So what does it do?
Reserpine binds irreversibly to VMAT and just shuts it down. So think through the consequences of that.
Well, the dopamine or norepinephrine gets synthesized, but it can't get into the vesicle for protection.
So it just sits there in the cytoplasm.
Where it gets chewed up by those mitochondrial enzymes we just mentioned.
And the nerve terminal eventually runs completely empty. It gets depleted.
So Reserpine doesn't just block the signal temporarily. It drains the whole system. It empties the tank.
It drains the tank. And clinically, that observation told us something incredibly profound about the biology of mood.
How so?
Back in the 1950s, when they used Reserpine for things like high blood pressure or psychosis, they found it caused severe, sometimes suicidal depression in a significant number of patients.
Because it drained their brains of serotonin and norepinephrine and dopamine?
Precisely. That terrible side effect was actually one of the first major clues that led to the monoamine hypothesis of depression, the idea that it's a chemical imbalance, specifically a deficiency of these neurotransmitters.
Wow. Okay, moving on. Target 3 is release.
This is the fusion event we talked about back in Lecture 2. Calcium comes in, binds to synaptotagmin, the snare proteins pull the membranes together, and the vesicle dumps its cargo.
And we can interfere with that.
Oh, absolutely. Botulinum toxin Botox works by literally cleaving those snare proteins. But for today, we're focusing on the small molecule drugs. So then we have target 4, which is receptor binding, the key fitting into the lock on the postsynaptic side.
And finally, target 5, termination.
The cleanup crew. This is arguably the most critical step for timing and for pharmacology. You have to clear that synaptic cleft to allow the next signal to be heard.
And there are 2 main ways to do it.
2 main ways. Reuptake, where you basically have a tiny vacuum cleaner that sucks the transmitter back up into the presynaptic terminal to be recycled.
Okay.
Or enzymatic degradation, where you have an enzyme floating right there in the cleft that chemically breaks the transmitter down.
And the slide contrasts reserpine with cocaine here to make a point.
It's the perfect contrast. While reserpine blocks storage, depleting the signal, cocaine blocks termination. Specifically, it blocks the reuptake transporter for dopamine.
It jams the vacuum cleaner.
It jams the vacuum cleaner. So the dopamine that gets released just stays in the cleft way longer, banging against the receptors over and over and over again.
So to use an analogy, reserpine turns the volume down to 0 because there's no music left to play. And cocaine breaks the volume knob off at full blast because the song just never ends.
That is a very vivid and a very accurate way to put it. This all ties into the concept of synaptic mimicry, mentioned at the bottom of the slide. It's the gold standard, really.
What does that mean?
It's the scientific standard for proving something is a neurotransmitter. If we, as scientists, take a micropipette and squirt that chemical onto a neuron, it has to produce the exact same physiological effect as stimulating the nerve naturally. It has to mimic the real thing.
It's the ultimate proof. Okay, so that's our general framework, the 5 targets. Now let's get into the specifics. Section 2 covers the hierarchical neurotransmitters, the amino acids. We are moving to slide 4.
Right. Slide 4 introduces us to glutamate, the primary excitator. If the brain is a hierarchical machine, glutamate is the electricity flowing through the wires. It's the main go signal in the cortex and spinal cord.
And the visual here is really interesting. It's titled the glutamate-glutamine cycle. It doesn't just show 2 neurons. It shows the presynaptic neuron, the postsynaptic neuron, and an astrocyte.
Yes. And this is a crucial detail that often gets left out of the introductory cartoons. We like to think of a synapse as just 2 neurons talking to each other.
But it's not.
In reality, it's what we call a tripartite structure. You have the presynaptic terminal, the postsynaptic density, and the astrocyte wrapping its little processes all around them like a glove.
So what is the astrocyte actually doing? It's not just structural support, is it?
Oh, far from it. It's the housekeeper. It handles the cleanup. See, glutamate is potent stuff. It's toxic in high concentrations. You do not want it lingering in the synaptic cleft.
So the astrocyte sucks it up.
But here's the key point. Unlike other transmitters, we don't just break it down in the cleft. And the neuron itself isn't the primary one doing the reuptake.
Wait, why not? Why doesn't the neuron just take it back? That seems more efficient.
Because having high concentrations of free glutamate inside the nerve terminal cytosol is actually dangerous for the neuron itself. It needs to be carefully compartmentalized. So the astrocyte steps in to help. It has these incredibly high affinity pumps on its surface called excitatory amino acid transporters, or ETTs. They are powerful vacuum cleaners for glutamate.
Okay, so it vacuums it up. Then what?
Then, inside the astrocyte, an enzyme called glutamine synthetase does something brilliant. It converts the glutamate into glutamine.
And what's the functional difference there? Why the conversion?
Glutamine is electrophysiologically inert. It doesn't excite neurons. It's safe. It's like putting the safety cap back on an explosive.
Ah, see.
The astrocyte then shuttles the safe glutamine back to the neuron. The neuron takes it up, uses an enzyme called glutaminase to pop the safety cap back off, converting it back to glutamate, and then immediately repackages it into vesicles for the next release.
That seems like a very roundabout process.
It seems convoluted, but it's about safety and energy conservation. Making glutamate from scratch, from glucose via the Krebs cycle, is metabolically expensive. This recycling system is key for the brain, which already consumes a huge amount of the body's energy.
But there's a dark side to this, isn't there? The slide mentions excitotoxicity.
There is. This is the clinical correlate. And it's what happens when this elegant cycle fails.
Like in a stroke.
A stroke is the classic example. Or severe ischemia or brain trauma. In those conditions, the pumps that drive this reuptake cycle fail.
Why?
Because pumps run on ATP, They're active transporters. If blood flow stops, oxygen delivery stops. If oxygen stops, ATP production in the mitochondria stops. The EAT pumps just die.
So the vacuum cleaner turns off.
The vacuum cleaner turns off, but the glutamate is still being released from dying cells, so it accumulates in the cleft to toxic levels.
And it just keeps hammering on the postsynaptic receptors.
Overactivating them, allowing a massive uncontrolled flood of calcium into the postsynaptic cell.
And we know from lecture 2 that calcium is a powerful signal, but in excess.
In excess, it is a poison. It activates a whole cascade of destructive enzymes, proteases that chew up the cytoskeleton, lipases that dissolve the membrane, endonucleases that chop up the DNA. The neurons literally excite themselves to death.
That's excitotoxicity.
That's excitotoxicity. It's a tragic irony. The very chemical that allows us to think and learn and function is what kills the cell when it's not tightly controlled.
That is genuinely terrifying. It really highlights why time is brain and stroke management. Okay, let's look at the receptors that are mediating this. Slide 5 gives us the diagrams for the two main ionotropic glutamate receptors, MPA and NMDA.
Right. These are the two you absolutely have to know for the boards and for understanding how the cortex works. Let's start with AMPA receptors.
Okay.
Think of AMPA receptors as the workhorses. They are simple, fast, and reliable. Glutamate binds, a channel opens, sodium rushes in, potassium rushes out. Boom. You get a fast depolarization.
So this is your standard day-to-day synaptic transmission.
This is what you're using right now to understand my words. If I want to tell you the sky is blue, I'm using billions of AMPA receptors.
Simple enough. But the NMDA receptor, the slide calls it a coincidence detector. That sounds much more complex, like a logic gate in a computer.
It is arguably the most sophisticated machine in any cell membrane. It's beautiful. Look at the diagram on the slide. Do you see that magnesium ion, Mg2 plus AUDA, sitting right in the middle of the channel pore?
Yeah, it looks like it's plugging the hole, like a bouncer blocking the door at a club.
A very stubborn bouncer. At the normal resting membrane potential of, say, negative 70 millivolts, the inside of the cell is negative. That negative charge electrostatically pulls the positively charged magnesium ion deep into the pore, plugging it solid.
So even if glutamate binds, nothing happens.
Nothing happens. The channel won't open because that magnesium plug is stuck right there in the way.
So how do you get rid of the bouncer?
This is the genius of it. You need two things to happen at the exact same time, a coincidence.
Okay.
First, you need glutamate to bind. That's the chemical signal. Second, you need the membrane to already be depolarized, usually because a whole bunch of nearby AMPA receptors have been firing away repeatedly.
So the cell has to be excited already.
Yes. When the membrane depolarizes, the inside of the cell becomes less negative, more positive. And that positive charge electrically repels the positively charged magnesium ion. It pops it out of the channel like a cork from a champagne bottle.
So the cell has to be listening, meaning it's depolarized, and receiving, meaning glutamate is binding at the very same time.
That is it. It detects the temporal association between two events. It's a Hebbian learning rule built into a single molecule.
And when it does open, what's different about it?
When it opens, it doesn't just let sodium in. Critically, it allows calcium to enter the cell. And as we just said, calcium is a powerful intracellular messenger. In this controlled context, it's not toxic. It activates signaling pathways that lead to long-term potentiation, or LTP.
Which is the cellular basis of learning and memory.
The cellular basis of memory. The NMDA receptor is essentially saying, hey, this synapse was active at the exact same moment that the whole cell was active. This must be an important connection. Let's strengthen it. That's how we learn.
That is absolutely fascinating. The slide also lists some key pharmacology here: ketamine, PCP, and memantine. These are all listed as NMDA antagonists.
They are. Ketamine and PCP, phencyclidine, are channel blockers. They go in and plug that pore, much like magnesium, but more permanently. By shutting down this coincidence detection system, they cause a dissociative state.
What does that mean exactly?
It means the brain stops processing the normal relationships between stimuli. Your sensory input gets disconnected from your perception of it. You are there, but you aren't really connected to what's happening. It functionally disconnects the cortex from the thalamus.
And what about memantine? That's an Alzheimer's drug. That seems counterintuitive. If NMDA receptors are crucial for memory, why would you block them in a disease of memory loss?
That is an excellent question. It's a common point of confusion.
Yeah.
The key is the difference in mechanism. In Alzheimer's, we think there's a low-level chronic leakage of glutamate. It's like a slow-motion excitotoxicity. Memantine is what we call an uncompetitive or low-affinity channel blocker. Think of it as a lunar plug. It's just strong enough to block that low-level pathological background noise, the toxic leakage.
But not strong enough to stop a real signal.
Exactly. When a strong physiological signal comes along, a burst of glutamate from trying to form a real memory, it's enough to push the memantine out of the way, so it filters the toxic noise, but still allows the important signal to get through.
Brilliant. It's a smart drug. Okay, let's flip the coin. Slide 6 takes us from the gas pedal to the brake. We're talking about GABA, the primary inhibitor.
Yes, GABA. And what's so elegant, as the visual on the slide shows, is that GABA is actually made from glutamate.
It's made from its own opposite.
It is. The enzyme glutamic acid decarboxylase, or J, simply clips a carboxyl group off of glutamate, and that one chemical tweak turns the brain's main go signal into its main stop signal.
Talk about efficient recycling. And just like glutamate, it seems to have different receptor types. Slide 6 lists GABA and GABA-B.
Right. GABA is the fast one. It's an inotropic receptor. It's a ligand-gated chloride channel.
So when GABA binds, chloride flows in.
Exactly. And remember your Nernst potentials. Chloride is high outside the cell, low inside. So when that channel opens, chloride, which is negatively charged, rushes into the cell. This hyperpolarizes it, pushing the membrane potential down to -75 or -80 millivolts, much further away from the threshold for firing.
It makes it harder to fire an action potential.
Much harder. And this receptor is the site of action for some very, very famous drugs.
I see them listed, benzodiazepines and barbiturates.
The most famous anxiolytics and sedatives, drugs like Valium, Xanax, and the older barbiturates, they bind to the GABA receptor at different allosteric sites. They don't open the channel themselves.
They just help GABA do its job better.
They make GABA better at opening it. They enhance inhibition. That's why they're sedating, why they reduce anxiety, and why they can stop seizures. They're just turning up the brakes all over the brain.
Okay, so that's GABA. What about GABA-B?
GABA-B is the slower metabotropic version. It's G-protein coupled. Instead of being a channel itself, it acts via a geoprotein to, among other things, open potassium channels.
And potassium is positive, so it flows out.
Positive charge flows out of the cell. The end result is the same, hyperpolarization, inhibition, but it's a slower, more prolonged effect.
And the drug mentioned here is baclofen.
Baclofen. It's a muscle relaxant we use for spasticity in conditions like multiple sclerosis or cerebral palsy. It targets these GABA-B receptors, especially in the spinal cord, to dampen the excessive motor neuron firing that causes the muscle tightness.
And termination for GABA.
It's primarily reuptake via a transporter called GAT1, but once it's back in the cell, it can be broken down by an enzyme, GABA transaminase or GABA-T. We'll come back to that when we talk about anti-epilepsy drugs later in the course.
All right, that's a great overview of the amino acids, the fast hierarchical data wires. Section 3 now brings us to slide 7 and a bit of an outlier, acetylcholine.
Acetylcholine, or HA. Yes, this is the exception to the rule we've just been establishing.
How is it an exception?
Well, every other major small molecule neurotransmitter we're going to discuss, glutamate, GABA, dopamine, serotonin, norepinephrine, relies primarily on reuptake to clear the synaptic cleft.
The vacuum cleaner method.
The vacuum cleaner method.
Yeah.
Acetylcholine does not. It is the only major one that relies almost exclusively on enzymatic degradation right there in the synapse.
And the visual shows the enzyme responsible, acetylcholine esterase, or ACE, just floating in the cleft.
And it is an astonishingly efficient enzyme. It has one of the highest turnover numbers known in biology. It can chop up a molecule of acetylcholine into choline and acetate in a fraction of a millisecond. It is a molecular Pac-Man.
Why the different strategy? Why not just use reuptake like everyone else?
Think about where acetylcholine is most famously used at the neuromuscular junction, the connection between nerve and muscle. For muscle control, you need the signal to be incredibly crisp and fast. Contract. Stop. Contract again. Stop again. You cannot have the transmitter lingering in the cleft, causing unwanted twitches. Degradation is simply faster and more definitive than reuptake for this specific purpose. It ensures one-to-one fidelity between a nerve spike and a muscle twitch.
But we can manipulate this system. The slide mentions HE inhibitors like donepezil and rivastigmine.
We can. Those are two of the main drugs we use for Alzheimer's disease.
How to help?
Well, in Alzheimer's, one of the key pathologies is the loss of cholinergic neurons that originate in the basal forebrain, specifically a place called the nucleus basalis of Meinert. These neurons are critical for attention and memory formation.
So you're losing the source of the HE.
You can't replace the dead neurons, but you can try to maximize the signal from the ones that are still surviving.
By killing the cleanup crew. By stopping the breakdown.
Precisely. By inhibiting the ACE enzyme, you let what little acetylcholine is released dwell in the synapse for longer. You increase its residence time, increasing the probability that it will find a receptor and have an effect.
But acetylcholine isn't just in the brain, is it? It's all over the body.
And that is the central problem with these drugs. ACE is the primary transmitter of the parasympathetic nervous system. It acts on the gut to increase motility, on the bladder, on the heart to slow the rate.
So you get side effects.
You get a lot of side effects. When you give these drugs for dementia, you often get nausea, diarrhea, urinary incontinence, and a slow heart rate, or bradycardia. You are boosting the rest and digest system everywhere. This is why the clinical rule is always start low and go slow.
That connects the pharmacology perfectly to the systemic anatomy. Okay, let's move into the next big category. Section 4, the diffuse systems, starting with slide 8, catecholamine synthesis.
Right. Now we're into the biogenic amines. These are the mood regulators. And chemically, they're all part of the same family. They are cousins.
The slide shows it really well. It looks like a biochemical assembly line.
That's exactly what it is. And it all starts with a single amino acid, tyrosine, which you get from your diet. The first step on this assembly line is the most important one.
Okay.
An enzyme called tyrosine hydroxylase, or TH, converts tyrosine into a molecule called DOPA.
And why is that step the most important one?
It is the rate-limiting step. It's the bottleneck. The neuron can be swimming in tyrosine, but it can only make dopamine as fast as its tyrosine hydroxylase enzymes can work. This step is highly regulated by things like phosphorylation and end product inhibition.
So if you want to shut down the whole factory, you don't mess with the workers on the line, you go straight to the foreman.
You block the foreman,
Yeah.
and we have a drug that does exactly that, metarosine. It's a potent inhibitor of tyrosine hydroxylase.
And where would you use that?
In a rare condition called pheochromocytoma. It's a tumor of the adrenal medulla that just pumps out massive life-threatening amounts of adrenaline. Metarosine is used to shut down the factory floor and get the patient's blood pressure under control before surgery.
Got it. So after DOPA, you get dopamine.
Right. And here's a really fascinating nuance of cell biology shown on the slide. Dopamine is made in the cytosol, just the general fluid of the cell. It's then pumped into the vesicle by our old friend, VANT.
The same transporter that Reserpine blocks?
The very same one. But here's the trick. If you're a neuron that wants to make norepinephrine, or NE, the enzyme that converts dopamine to norepinephrine, it's called dopamine beta hydroxylase, is not floating around in the cytosol.
Where is it?
It lives inside the synaptic vesicle.
No way. So you have to pack the dopamine into the vesicle first, and then it gets turned into norepinephrine.
Correct. The vesicle itself is a little chemistry lab, which means if you block VMAT with a drug like Reserpine, you don't just lose your ability to store dopamine. You completely stop the synthesis of norepinephrine because the raw material never reaches the enzyme.
That is such a cool detail. It explains why a drug like Reserpine hits both of those systems so hard.
It's a 2-for-1. Okay, moving to slide 9, let's focus on dopamine specifically. This is the celebrity neurotransmitter. It gets all the press.
And the slide shows 3 key pathways in the brain that we need to know.
Understanding these 3 pathways is the absolute key to understanding both psychiatry and the side effects of neurologic drugs. Pathway 1, the nigrostriatal pathway.
That projects from the substantia nigra up to the striatum.
Yes, part of the basal ganglia. This is the motor pathway. It's what helps facilitate smooth, voluntary movement. When you lose the dopamine neurons in this pathway, you get Parkinson's disease.
The rigidity, the tremor, the slow movement.
All of it. Now, pathway 2, the mesolimbic and mesocortical pathways.
From the midbrain out to the limbic system and the cortex.
This is the famous reward pathway. It's what gives you that feeling of pleasure and motivation. It's what makes you feel good when you eat chocolate or win a bet. But it's also the pathway implicated in psychosis and schizophrenia. It's thought to be overactive in those states.
The same pathway for reward and psychosis. That's tricky.
It's very tricky. And finally, pathway 3, the tuberoinfundibular pathway.
That's a mouthful.
It is. It just means it connects the hypothalamus down to the pituitary gland. And in this pathway, dopamine's main job is to act as a brake. It tonically inhibits the secretion of the hormone prolactin.
Okay, so let's connect these pathways to the pharmacology on the slide. It highlights the D2 receptor.
The D2 receptor is the main target for almost all antipsychotic drugs, what we used to call neuroleptics. If you have a patient with schizophrenia who's having hallucinations and delusions, the goal is to block D2 receptors in that mesolimbic pathway.
But the drug doesn't just go there, it goes everywhere in the brain.
And that is the tragedy of the older, typical antipsychotics like haloperidol. You give the drug to treat psychosis, and yes, it blocks D2 receptors in the reward pathway, but it also blocks D2 receptors in the nigrostriatal motor pathway.
Where you need them for movement.
And so you induce a drug-induced Parkinsonism. You cause what we call extrapyramidal symptoms, or EPS. You create a movement disorder in order to treat a thought disorder.
And what happens if you block them in that pituitary pathway?
You take the brake off prolactin, so prolactin levels in the blood shoot up. That's called hyperprolactinemia. And clinically, the patient can start actuating, that's galactorrhea, and have amenorrhea or other reproductive issues.
Wow. So this is a perfect example of why knowing the anatomy, the where, is just as important as knowing the chemistry, the what.
You can't separate them. You aren't just targeting a receptor, you are targeting a receptor in a specific place.
The slide also mentions cocaine and amphetamine again here in the context of termination.
Right. So dopamine is normally cleared from the synapse by the dopamine transporter, or DEET. Cocaine is a straightforward reuptake inhibitor. It binds to DEET and physically blocks it. Dopamine builds up.
But amphetamine is different, you said.
Amphetamine is much more aggressive. It's sneaky. First, it gets transported into the nerve terminal by DEET like a Trojan horse. Then it gets into the vesicles via VMAT and causes them to dump their dopamine out into the cytosol.
Okay.
And then it reverses the direction of the DEET transporter itself, causing it to pump all that dopamine out of the neuron and into the synapse.
So it's not just jamming the vacuum cleaner, it's turning it into a leaf blower.
An excellent analogy. It causes a massive, non-activity dependent flood of dopamine release.
No wonder it's so potent and has such a high abuse potential. Okay, let's move on to dopamine's cousin on slide 10, norepinephrine, or NE.
Norepinephrine is the neurotransmitter of arousal, vigilance, and mood. And the visual on the slide is classic. It shows the locus ceruleus, this tiny little blue spot deep in the brainstem.
It looks so small on the diagram, but the arrows coming out of it go absolutely everywhere.
That is the very definition of a diffuse modulatory system. These few thousand neurons project to the entire cerebral cortex, the cerebellum, the spinal cord.
Yeah.
Everywhere. They are what wake up your brain in the morning. They're central to the fight or flight response.
The slide makes a special point to highlight the alpha-2 receptor here. It calls it an autoreceptor.
Yes, the autoreceptor concept is fundamentally important for pharmacology. It's a negative feedback loop. The alpha-2 receptor sits on the presynaptic terminal.
The one sending the signal.
Right. When norepinephrine is released into the synapse, some of it floats back and binds to this alpha-2 receptor on the terminal it just came from.
And what does it tell the terminal?
It basically says, okay, that's enough. We have plenty of signal out here. You can stop releasing now. It's coupled to a J protein, which then inhibits calcium channels and stops further vesicle fusion.
It's a thermostat for neurotransmitter release.
A perfect description. And of course, we have drugs that can hack this thermostat. Clonidine, for example, is an alpha-2 agonist. It stimulates this receptor.
So it tricks the neuron into thinking there's too much norepinephrine out there.
And the neuron shuts down its own release. That's how clonidine lowers blood pressure and can be used for anxiety or sedation.
And on the flip side, the slide mentions an antidepressant, mirtazapine.
Mirtazapine does the opposite. It's an alpha-2 antagonist. It blocks the autoreceptor. It cuts the brake line.
So the neuron never gets the stop signal.
It never gets the stop signal, so it just keeps pouring out more and more norepinephrine, and through other mechanisms, serotonin as well. It's a clever way to boost the signal by disabling the safety mechanism.
And for termination, reuptake again?
Reuptake via the norepinephrine transporter, or NET. This is the primary target of the older tricyclic antidepressants, or TCAs, and the more modern SNRIs, like venlafaxine or duloxetine. They block the reuptake, keeping NE active in the cleft to treat depression and also neuropathic pain.
Okay. Slide 11 brings us to the last of the big monoamines. Serotonin, or 5-HT, the mood stabilizer.
Serotonin is fascinating, mainly because of its incredible receptor diversity. The slide notes there are over 14 different subtypes of serotonin receptors.
That seems excessive. Why so many?
It allows for an incredible degree of nuance in cellular signaling. Most of them are metabotropic, G-protein coupled, affecting the cell state slowly. But there is one major exception, the 5-HT3 receptor. It is the only one that is an iotropic channel, a ligand-gated cation channel.
And that one is linked specifically to nausea and vomiting.
Yes. 5-HT3 receptors are found in high density in the gut and in the brain's vomiting center in the brainstem. That's why drugs like ondansetron brand name Zofran are so effective. They are 5-HT3 antagonists. They block that specific receptor to stop nausea and vomiting, especially from chemotherapy.
But the other receptors, like 5-HT1A or 5-HT2A, are the ones involved in mood and anxiety.
And psychosis and even hallucinations. Psychedelic drugs like LSD are potent partial agonists of the 5-HT2A receptor, for instance.
And termination is, once again, reuptake, this time via SERT. the serotonin transporter.
Right. And SERT is the target of the most commonly prescribed class of psychiatric drugs in the world, the SSRIs, selective serotonin reuptake inhibitors.
Like Prozac and Zoloft.
Fluoxetine, sertraline, all of them. They do one thing, and they do it well. They selectively block that SERT vacuum cleaner, boosting the amount of serotonin in the synaptic cleft to hopefully alleviate depression and anxiety.
Slide 12 seems to wrap up this section with the volume transmission concept we touched on at the very beginning.
It's just so important, it's worth reiterating. These monoamines, dopamine, norepinephrine, serotonin, don't always act in a point-to-point fashion. They often spill out of the synaptic cleft.
And they diffuse.
They diffuse to affect receptors that could be microns away.
Okay.
It's not a private whisper between two neurons. It's a public announcement broadcast over a whole region of the brain.
The slide uses that great analogy again, a sprinkler system versus a garden hose. It's the best way to think about it. And this is why these systems regulate state. If you dump serotonin, or norepinephrine, over a huge area of the cortex, you shift the mood and attentional capacity of the entire machine, not just the processing of one specific thought.
It provides the emotional context for our cognitive content.
Beautifully put.
Okay, that brings us to our final section, Section 5, Integration and Clinical Correlates. Slide 13 gives us a nice summary table of enzyme inhibitors.
Right. This is a bit of a matching game for the boards, but it's life-saving knowledge in the clinic. We can divide them into two groups.
Synthesis blockers and breakdown blockers.
Exactly. For synthesis, we already mentioned metaricine blocking tyrosine hydroxylase for pheochromocytoma. For breakdown, the big ones are the MAO inhibitors.
The monoamine oxidase inhibitors, these were the first antidepressants, weren't they?
They were. By stopping the enzyme that breaks down all three monoamines, you increase the stores of dopamine, norepinephrine, and serotonin all at once. It's a shotgun approach.
But very effective, especially for what we call atypical depression. But they're also dangerous because of their interactions with certain foods, like aged cheese and red wine that contain a substance called tyramine, the infamous cheese effect.
Right.
It can cause a hypertensive crisis.
And we also see the ACE inhibitors here, again, for Alzheimer's. And a new one I see is a GABA transaminase inhibitor.
Vigabatrin. We mentioned that GABA can be broken down by the enzyme GABA-T. Well, vigabatrin stops that breakdown.
To get more GABA.
More GABA means more inhibition. So it's used as an anti-epilepsy drug, particularly for a severe type called infantile spasms. It's all about calming the electrical storm in the brain.
The logic is consistent. Okay, slide 14 summarizes the other side, the transporter blockers.
We've covered most of these pretty well already. Cocaine is the dirty drug. It hits everything. NET, SERT, and DAT.
Whereas the modern drugs are more selective.
The whole history of psychopharmacology is a quest for selectivity. SSRIs just hit SERT. TCAs are a bit messier, hitting NET and SERT, plus a bunch of other receptors that cause side effects.
And gate 1, I see that listed.
Tiagabine. It's another anti-epilepsy drug. It blocks GACI1, the GABA reuptake transporter.
So again, the logic holds. You block the reuptake of an inhibitory neurotransmitter, and you get more inhibition.
More inhibition, fewer seizures.
All right. Finally, slide 15, the conclusion. The neuropharmacologic matrix.
This is the big picture. This is where we synthesize everything we've talked about for the last hour. We can divide the central nervous system's chemical signaling into a 2x2 matrix.
On one axis, you have hierarchical versus diffuse. And on the other axis, you have excitatory versus inhibitory.
Exactly. So let's take the hierarchical systems, glutamate and GABA. Their job is to determine what happens.
They transmit the specific data, the yes or no.
Right, the action potentials. So drugs that act on these systems, general anesthetics, anti-epileptics, sedatives, they tend to have global effects. They turn the whole system on or more often off. They control consciousness and seizures.
They control the existence of the signal.
Yes. They're state-dependent, but they aren't specific in terms of mood or content. Now, contrast that with the diffuse systems.
Acetylcholine, dopamine, norepinephrine, serotonin. Their job is to determine how well it happens.
They modulate. They set the tone.
Yeah.
So drugs that act here, antidepressants, antipsychotics, stimulants, they don't usually put you to sleep, though they might make you drowsy. They change your mood, your perception, your motivation. They treat the quality of the conscious experience, not the existence of it.
That is such a profound and useful distinction. It perfectly explains why we don't treat clinical depression with Valium, which is a GABA drug, or
Right.
treat epilepsy with Prozac, which is a serotonin drug.
You have to match the tool to the system. You have to know if your job is to fix the fundamental wiring or just adjust the volume and the tone.
Well, we have successfully unpacked the chemical software of the brain. To recap, we trace the entire life cycle of a neurotransmitter from synthesis to termination. We saw the crucial distinction between the fast, point-to-point hierarchical systems of glutamate and GABA,
And the slow, modulating, sprinkler-like diffuse systems of the monoamines.
and we connected all of these basic science mechanisms to the drugs we use every single day in the clinic.
It's the toolkit. And now that you have it, next time, in Lecture 4, we're going to zoom way, way out. We've been looking at the molecules and the synapses. Next, we look at the machine itself.
We're finally covering macroscopic organization and embryology.
We'll see how these tubes and vesicles and chemicals actually organize themselves into a recognizable brain.
From the microscope to the MRI. I absolutely cannot wait. But before we sign off, here's a final thought for you to chew on. We spent a lot of time today talking about volume transmission neurotransmitters like serotonin and dopamine spilling out into the extracellular space like a sprinkler system to set our overall mood and personality. If so much of who we are, our happiness, our anxiety, our drive, is determined not by precise wiring, but by a chemical soup just sort of sloshing around in the brain, how much of us is actually precise architecture and how much is just humidity?
That is the question that keeps neuroscientists and philosophers awake at night.
Thanks for listening to this deep dive into neuropharmacology. We will see you for Lecture 4.